Yesterday afternoon, I just realized that Nanosafety was the start of today's lecture. And I wondered why. And maybe it's very simple, because if consumers don't perceive nanotechnology to be safe or safe enough, there will be no great demand for the products. The present researchers within Nanosafety come from 3 faculties and several departments. The most active ones are listed here. And we have tried to define 3 overarching themes for our work, from workplace emissions to physiological effects, from the molecular mechanisms to effects on living organisms, and from mode of action to the environmental and societal impacts. Maybe this is your picture. The nano developers, like the hair, taking great leaps in different directions. Sometimes it might be a dead alley, but they are always energetic and always full of new ideas. And then the safety people, on the other hand, keep saying, be careful, take it slowly. However, there is a second story about the hare and the tortoise, and the xenon paradox. Because the tortoise in this story starts with an advantage. And we do have an advantage. For instance, for the workplace, it is the precautionary principle to use safety and protection devices to protect the work environment. And if that is not enough to make it safe, you have to protect the worker. But this isn't enough, because you have to consider safety not only during production, but in a much broader perspective. What about the production of the raw materials? What about worker safety in the production of raw materials? And what about the life cycle of the product, the environment, or the persons that actually are involved in recycling or deposition? That is questions that has to be with you all the time. But what are we doing? We are looking at workplace exposure to manufactured nano-objects, measuring exposure mostly in air, but also on surfaces, in the lab, in the nanolab, but also during small-scale production, looking at carbon nanotubes at the small-scale producer, looking at emissions during maintenance work of quite normal particle generators for metal nanoparticles, or at nanowires at a small-scale producer. You may find a poster on that later today. And why? Why do we try to measure in detail in workplaces? Well, one reason for that is actually to be able to reproduce the real exposure setting in an experimental situation, and vice versa. Because we need to develop relevant exposure systems for the environmental studies. The dominant role of the nanoparticles to enter the body is through the lung. And the lung is essentially an air-liquid interface, as can be seen at the top panel. So, a system to actually expose liquids to nanoparticles, one by one, has been developed. You can see a schematic picture of that at the bottom. So here, particles can be deposited on a liquid with or without cells. So far, this has been used for metal nanoparticles. The next to come is to use it for nanowires from air taxi, and that is much more difficult, and we have not solved that yet. But the question is not only about finding a good model to exposure in the lab. You also have to have a good system to look at effects. And for that, we do have an advantage. We have an advantage, for instance, from air pollution research. We clearly know that the risk for heart attacks or stroke is higher on days with high particulate levels, even in Scania. And if you take myocardial infarction, that has to do with coagulation, a blood clot in the vessels of the heart, or stroke, a blood clot in the vessels in the brain, or it could be bleeding. So clearly, coagulation is affected by very small particles. And now I have to explain, because coagulation is about activation and inactivation of proteins in a cascade. Like the yellow protein interacts with the red protein, which interacts with the orange, with the green, and so on. And then there is blue protein coming from the side and disturbing this. And this is actually the coagulation cascade, this delicate balance. So, likely, coagulation could be a good model system to look at small particle effects. And another thing to be explained is when you have a protein particle in the bloodstream and it very rapidly is covered with proteins. So, if you put these two together, you can clearly imagine that nanoparticles coated with the proteins might disturb what is happening in the balance of coagulation or no coagulation. And this has been used in a model system. Small plastic nanoparticles affected coagulation, but it differed between the very small ones and the not so small ones. With very small ones, there was an inactivation of coagulation. And with the larger small ones, the coagulation was activated. So clearly, size matters. And then, of course, we could remember that this is a model system. It doesn't tell us what is bad or good. The forming of a blood clot is good if I cut my hand, but it's bad if it happens in my brain. But the point is that size matters. But to make it more complicated, this only happened with negatively charged particles, so we have to understand more. For fiber-like particles, we have another advantage. As a young medical doctor, I met many hundreds of asbestos workers. All of them were worried because of health effects of their work. Some of them had severe respiratory diseases. Some of them died because of an asbestos-related cancer. But that asbestos tragedy also led us to investigate a lot about fiber toxicity. And we do know from that size, shape, persistence, and surface matters. And this knowledge is, of course, very relevant when it comes to long, thin, and persistent objects of other kinds than asbestos, for instance, nanotubes. I looked for an old picture, like pictures looked alike in the early 1980s, and I found a macrophage, a scavenger cell, trying to destroy an asbestos fiber in the lung. And the result is what we call frustrated phagocytosis. To put it very simple, I try to eat it and it hurts. The model system for nanowires uses fruit flies and small nanowires can be injected into the fruit fly, as on the top panel. And then small nanowires are internalized by immune cells, like in the second panel, and then it is possible to study what happens in the immune system, signals, recognition, and so on. But if the injected nanowires are too long, the result will be frustrated phagocytosis, as in the third picture. The new thing is to actually use fluorescently barcoded nanowires. And then it is possible to actually look at very distinct nanowires with different surfaces and see what happens. The next thing is to go not only for small things one by one, but also together. And here you have a fish tank. The fish at the left-hand side has normal food, normal Daphnia. And the fish on the red-hand side, they get food with nanoparticles in them. And they move much more slowly. They don't eat. And as you can see, they stick to the bottom. So not only the single fish, but the interplay between fish is different. Oh, sorry. Is this relevant? Yes, it might be relevant also outside the fish tank. There is a lot of plastic disintegrating in the ocean. But there is a third thing. Please go on. There is a third story about a tortoise and a hare. And that story, with the fast-running hare and the slowly moving tortoise, ends differently. It ends in collaboration. And maybe that would be the really great advantage.